So the Nobel Prize this year was awarded to three individuals, Shimon Sakaguchi, Mary Brunkow, and Fred Ramsdell, and for their fundamental discoveries in concerning peripheral immune tolerance. And I'll speak a lot more about this so you understand what I'm talking about. So essentially, these, again, to reiterate, these are the three individuals. Mary Brunkow works at the Institute for Systems Biology now, and Fred Ramsdell at Sonoma Biotherapeutics. But at the time of these fundamental discoveries were happening, they were working in other places. Um, Shimon Sakaguchi has remained at Osaka University for a long time now. Why is this discovery important? I mean, the Nobel Prize is typically awarded to discoveries that change our fundamental understanding of some phenomena and expand our comprehension of things or mechanisms that we don't know about, right? In ways that change the entire paradigm of study, right? And so how does it expand our comprehension of the immune system, right? So just to speak about the immune system a little bit, right? The immune system is made up of multiple components. It comprises multiple organs and cellular cells and tissues. The organs include the thymus, the spleen, the lymph nodes in the various parts of the body, and there is a cellular component as well, I think. And this cellular component comprises many other types of cells as well. It includes white blood cells, obviously, but different types of cells as well. And this cellular component arises from what we call hematopoietic stem cells. Right? These are progenitor cells, and these hematopoietic stem cells reside in the bone marrow. Right? And the hematopoietic stem cells essentially have the what we call totipotent. Right? They can give rise to any kind of cell in the body, really. And so they give rise to two lineages. One is called the myeloid lineage, and the other one is called the lymphoid lineage. The myeloid lineage gives rise to cells that are involved in non-specific immune attacks on foreign bodies, but the lymphoid lineage gives rise to what we call the part of the immune system that actually has memory, that can remember a problem, an invader, a foreign body, and mount an attack many years in the future. So it retains its. So this comprises like T cells and B cells, right? And B cells synthesize antibodies against antigens that have occurred in the host in the past, and T cells can also mount direct attacks on these foreign bodies, right? Whereas the myeloid component gives rise to cell. So there's an error here. This should say myeloid progenitor. Okay, so you can have dendrites, monocytes, neutrophils, basophils. These will give rise to CD4 cells and CD8 cells. Right. So CD4 cells are essentially what we abbreviate them, or they're known by the name cytotoxic T cells, and CD4 cells are the helper T cells, and the CD8 the cytotoxic T cells. This will become a little clearer to you as I go along, right? But these are like essentially what we're sort of interested in, and but this is just to sort of highlight like the complexity or the variety of cells that these two lineages give rise to, all of which are involved in fighting, you know, attacking pathogens, foreign bodies, and so on. There is a problem though. The problem is that the immune system is also constantly evolving. So every time there's a new insult that it receives, that the host receives, the immune system learns, identifies the pathogen and the part of the pathogen causing the problem, and it creates antibodies against it and a class of T cells that will remember what that antigen was. So in the future, as soon as it hits, again, it'll launch a even bigger attack. And essentially, it's also the basis for how vaccines work. Right? You create essentially a large repertoire of T cells that can actually remember and mount an attack in the future. And the immune system will evolve over the course of the lifetime of the individual. So there's no stopping it. The more the number of antigens that we come across, the more the immunity we possess. And you see this mentioned anecdotally when some of us go to countries which are free of a lot of diseases, right? So we actually find that we are more immune to what might be causing people problems there or infections there. So essentially, just to reiterate, the immune system protects against pathogens, and central to this ability is to tell self from non-self. How does the immune system know, in other words, what belongs to the host and what is off limits, and what doesn't belong to the host and what is foreign, right? And so that therein lies its ability to protect you. So it's a very thin line. If any component of the immune system goes rogue for any particular reason, and there can be any number of these, it can actually cause the immune system to go rogue. Right? And this is precisely what happens when we have an autoimmune disease. Yeah, come in, come. This is precisely what happens when you have an autoimmune disease. Right? And remember that cells and tissues and these organs, they all have a. I should also mention that any lineage, when I showed you the chart of the hematopoietic stem cells giving rise to the myeloid cells or the lymphoid cells, and then each of the myeloid and lymphoid cells giving rise to other types of cells, remember that each cell in the immune system, and for that matter in the entire body, has a developmental program that it adheres to very strictly, right? So a cell knows exactly what it's going to become, right? And so if there is and this developmental program is executed by the temporal and spatial, you know, deployment of certain or expression of certain genes in those cells. And if there's a mistake in the timing or spatial expression of those genes, something is off. And so there's no uh, shortage of the number of things that can go wrong. You know? And so and actually the impressive thing that is that it actually, in most cases, it actually works. So one way that the immune system maintains or develops this ability to distinguish or discriminate between self and non-self is by what we call central tolerance. So what happens in that is that the immune cells, imagine this organ, the thymus, and I think I have a picture here. So you have an organ called the thymus, and what happens is that the immune cells that are being synthesized, that are still immature, are passed through. So maybe I should like step back and talk about the T cells first. Essentially, this is how, when I spoke about how T cells maintain memory and can attack certain pathogen in the future, this is how it can do it, right? So if you have a cell that's infected by a virus, right, and what the cell does is exhibit the virus fragment on its surface, the T cell comes along, identifies that particular part of the cell of the virus, and then launches an SOS message that other T cells can listen to and come and kill that particular cell or neutralize that particular antigen. And so this is essentially the process. Now, a T cell is like a very large variety of T cells, each of which can identify and bind to one specific kind of part, or like that fragment that you're talking about. So it's a really vast variety of these T cells, because the if you've been exposed to a certain pathogen or an antigen or a foreign body, your T cells know about it, and they can recognize, and they'll have the requisite cell surface receptor that can bind to these fragments in the future, anytime in the future, right? And so, but these are what we call mature T cells, right? They've already been developed. Right? And there are two different kinds that are really important. One is the cytotoxic T cell that actually can directly identify a certain part on an infected cell and kill the cell. And the T helper cell, which can go and identify the part and release an SOS message that recruits other T cells and other parts of the immune system and causes them to come and to a certain spot and cause killing or destruction of it. If you've ever had been injured, if you've noticed any redness or swelling, that's essentially the immune system going into overdrive. It's causing inflammation and it's causing redness. It's there to fight that particular whatever is wrong. So this is a picture of a baby, but the thymus is what is important for the maturation and development of T cells. Right? And so in the thymus, the T cell is essentially made to pass through a quality assurance process. Right? And so if the T cell in the thymus is able to bind to, like, any protein or component of the human body, then it is eliminated because it is already rogue. So all the T cells that are released from the thymus into the bloodstream are essentially those that do not recognize any part of the human system, or recognize it, but recognize it as self, right? And so they don't attack, and that's how they're trained. And therefore, this quality assurance system makes sure that your immune cells don't recognize and go after self cells, and so as to preserve the integrity of the tissues of the host. Now, this is what we call central tolerance. However, when these experiments were done, they found that the central tolerance didn't really cover all the facts. There had to be something going on inside the bloodstream as well, closer to the site of injury, right? Where, and so these lines of evidence suggested that you couldn't just have the central immune systems, the central tolerance as its premise. You had to have something else also. So these are some of the experiments that Sakaguchi conducted. When he took the thymus, when he removed the thymus from 3-day-old mice, embryos or newborn mice, the expectation was that the mice will now have a much feebler immune system. However, they were in for a surprise. So what exactly happened was that the immune system went into overdrive and started killing these mice. Right? So they would find that these mice would really become really sick. Their organs would be really damaged, and they found that T cells were going and launching these attacks on the cells of the mice. So, in effect, removing the thymus was not making the immune system weaker; it was making it go rogue. Right? He did another experiment where he wondered. So, because they were finding a lot of these T cells in the vicinity of these organs that were damaged, he decided to take a healthy donor, which was genetically identical to the cells, to the mice that he did the first experiment in, and he isolated these T cells, and injected them into the mice that were becoming unhealthy, or the mice from which the thymus had been removed, and they found that they were protected. So obviously, this suggested to Sakaguchi that it wasn't just the central immune tolerance or the system of quality assurance in the thymus that was helping, but also at the level of the cellular component. Right? So it's not just at the organ, but also at the cellular level. And so essentially, these mice that don't have a thymus and receive these T cells are protected from autoimmune disease. Okay? And so Sakaguchi, but unfortunately, what happened was that, so there appears to be a cellular component in addition to the thymus that modulates the immune response. And so what class of cells are these, and how could their presence be demonstrated? They weren't sure then, right? And Sakaguchi did several more experiments to identify these as well. But here's what was not known, right? If I go back to the previous slide, and so it appeared, therefore, from this line of inquiry, that there had to be some class of T cells, in addition to the already known and documented T helper cells and cytotoxic T cells, that are actually engaging in an immune response. And so maybe a third kind. So when Sakaguchi actually spent like several more years trying to identify and separate and distinguish the various classes of T cells. One approach that he took was using the cell surface receptor. Now we know that T helper cell has a CD4, or a molecule called CD4 on its surface. It's like a name tag on its surface. Cytotoxic T cells have CD8, right? But he identified this new class of cells that have CD4 and CD25 on them. So essentially, the way he discovered this was when he injected regular CD4 cells into mice. What they should have done is helped evoke a larger immune response, because CD4 corresponds to T helper cells. But he found that instead of evoking a larger immune response, they caused a suppression. So obviously there was something else, some other class of cells that had infiltrated or sort of contaminated his group of cells that were not just T helper cells, but also something more powerful and whose presence was sort of masking the effect of the regulatory CD4 cells, right? And so these were cells that had not just CD4, but also CD25. And so, and so he set about trying to isolate these cells, this class of cells, and he identified eventually um, this class of cells called CD25. And if you just use the CD25 cells in mice that didn't have a thymus, it would still stop them from becoming sick. So essentially, it could, the CD4 plus CD25 cells, that cohort of cells alone or that class of cells alone could negate the autoimmune problem, right? And so that's when they identified that they agreed that there was a separate class of cells that was inhibiting the immune systems, or rather modulating the immune response in the peripheral blood system and causing it to not cause harm. And so this class of cells was called the T regulatory cells. And therefore, this is what would be called the peripheral tolerance system, right? And so, but a lot of scientists weren't convinced. Like, could you actually, did I get ahead of myself? Could you actually demonstrate the presence of these cells? Um, and was this a real thing? I mean, was it true that these T helper cells were actually protecting the cells or the mice from autoimmune diseases? The evidence that he presented didn't appear to be sufficient enough for a lot of scientists, and so uh, he continued working on it, but confirmation and help for his work came from a very different line of inquiry happening like far away, right? So this was the paper that Sakaguchi published, showing that this class of T cells existed and that this class of T cells was actually protecting from autoimmune diseases, right? And this was a paper, and but the evidence apparently was not sufficient for a lot of scientists, and so essentially this third kind that are called T regulatory cells bearing CD4 and CD25. And so the scene sort of shifted because Sakaguchi was obviously still working on this, but a different line of inquiry was happening in a different part of the world and in a very different area, essentially, right? So when the Los Alamos National Laboratory was set up to study the atom bomb or develop the atom bomb, and after the atom bomb was tested, a lot of interest went into identifying the effects of this radiation on humans or on animals. And so at the Oak Ridge National Laboratory, they were studying the effects of radiation on mice. But ironically, this particular mouse that is of interest came up as a result of a spontaneous mutation. Spontaneous mutations happen all the time, but it wasn't really the effect of radiation that caused the mutation, but just evolved spontaneously. And so that particular mouse was called scurfy, right? It's a spontaneous mutation that happens in certain mice strains, um, and it causes a certain type of phenotype. And by phenotype, I mean that it has a specific set of characteristics around it. It has red eyes, it has scaly, flaky skin, it has a unusually enlarged thymus, a spleen and lymph glands. And these mice are viable only for a few weeks. They don't live very long, so they're not very well. So when they looked at these mice, which is called scurfy, and so it appears that the scurfy, that phenotype is caused because a lot of T cells are attacking the cells of the mice, essentially, right? And so whatever mutation it was or is that is causing the phenotype is actually causing a rebellion in the immune system. And so the question they asked was, so this work was happening anyway at the laboratory, but Mary Brinslow and Fred Ramsdell were cognizant of these developments. And at that time, they were working in this company called Kairos Biosciences, and this company was involved in, you know, developing drugs for autoimmune diseases. So the question they asked was, I mean, will an investigation of these mice help in our understanding of autoimmune disease? And what is the mutant gene that causes scurvy essentially? Will it, this is a classic case of what we call forward genetics. You see a characteristic and you try to look for the gene that is causing it. Okay? As opposed to reverse genetics, where you have a set of genes which you can mutate and look at what effect it has. And so the question they asked was, what is the mutant gene that causes scurvy? I want to sort of step back and talk about, this is actually a gigantic task. I mean, especially in the 80s and early 90s, when our tools of molecular biology weren't good enough or weren't capable enough, you had to do this the old way, where you would have to cross different types of mice and then identify and map using the frequency of recombinants the positions of these particular genes on the genome, and it would take years. The same experiments can be done today in about 3 days, right? But it took them like years, you know. So that's testimony to their resilience and their perseverance. So they went about it. They decided that it has to be asked anyway, the question, and so we'll go ahead and like work on it. And so they did a large number of crosses, and they found that this particular mutation appears to be an X-linked mutation. In the sense, it's inherited in an X-linked manner. Now, we all know that humans have two sets of genes. Uh, Females will have two X chromosomes, which are sex chromosomes, and males will have one X chromosome. And so, if the mutation is present in only one of the X chromosomes, it will cause a problem in males, but females will only be a carrier because they just have one copy. For females to have the problem, they need to have two copies. So the likelihood of their having a problem is like much lesser. Right? And so they identified it as an X-linked gene, and then they narrowed down, and this might seem a little absurd in this era, but they narrowed down on a region of the gene, of the chromosome. It's about 500,000 bases in size. That's a lot. And a base is essentially one unit of the genome, right? So you have 500,000 units of those, and you're looking for the gene you want in that space, right? So they did a lot more experiments, and again, these are what we call mapping experiments. What you do is you cross mice that are mutants with mice that are wild type or normal, and you see the frequency of recombinants being formed. And from the numbers, because the frequency of recombination depends on its proximity to that particular gene, you can identify based on the frequencies what the position of the gene is relative to other genes. And so that's how they slowly, and they found that this particular region had about 20 different genes, and they worked on each one to see if mutating that particular gene would cause something. It was only the 20th gene that turned out to be the right gene. So it took them a long time there as well. And so, like I said, 500,000 base pairs and 20 genes. And this is again that 500,000 base pair region of the genome. And you can see each of these indicates one position or one locus on the genome where that gene exists. And this particular gene, the FOXP3 gene, is actually shown in an enlarged fashion. What the mutation does essentially is cause a truncation when the gene is expressing a protein. Right? So instead of getting a full-length protein, which would do its job properly, you would get a smaller protein that doesn't and cannot do its job properly. We call this a nonsense mutation. It is what we call a stop site, and then creates a dysfunctional protein. And so they did the other experiment also, right? So it's not just the mutation, the presence of mutation that causes a problem, but introduction of a gene, a normal gene, into a mutant organism should restore the phenotype, the normal phenotype, and they saw that it does. So when we do these molecular genetics experiments, we call it complementation. So if you have a mutation in a gene and it's causing a problem, you observe that first. You can map the problem to that particular mutation. But what gives you even more confidence in that discovery is that the introduction of a gene, of a normal gene, into a mutant organism should restore the normal phenotype. Right? And that's what they did. They found that when they introduced the transgene, which they call the transgene, it actually restores there's no problem in the mice anymore. So they did it both ways, and they showed that it is in fact definitively a problem in this particular gene that's causing a problem, right? And so we call it rescue essentially. So wild type FOXP3 gene, which is the normal gene, rescues male scurfy mice from disease. So that's essentially what they're saying. So essentially, after several years of research, the faulty gene was identified, and because of its similarity to an existing group of genes, it was called the forkhead box gene, right? That family of genes was called the forkhead box genes or FOX genes, and it was called FOXP3. Fortuitously, this was the paper where they presented this data. They said disruption of a new protein, a new forkhead protein, results in a fatal lymphoproliferative disorder of the scurfy mouse. You know, when researchers are working on one thing, they're obviously cognizant or aware of problems happening in other areas or other diseases in their area as well. And they found that they became aware of a particular autoimmune disease happening in humans, which had characteristics similar to those of the scurfy mice, right? Physiologically at least, if not in gross anatomy terms, right? And so this was called IPEX. So they wondered if the same mutation that's responsible for scurfy also is responsible for a rare X-linked autoimmune disease, IPEX, right? And in order to answer this question, they had to get samples of, because it's a rare disease, it's not common, and so they had to collate samples from all over the world, uh, samples from boys who were afflicted by this disease, and they found that it was indeed there was indeed a mutation in the human equivalent of the FOXP3 gene in these patients. And so this was when they presented this information, this study, the X-linked neonatal diabetes mellitus enteropathy and endocrinopathy syndrome, which is essentially the full form of IPEX, right, is the human equivalent of mouse scurfy. So they could tell that the causal gene is the same gene, and it's causing a problem in humans as well, right? And so once this happened, there was obviously, these were contemporaries like Mary Brinslow and Sakaguchi and Fred Ramsdell. Sakaguchi immediately understood that this could actually be the reason why the T cells become dysfunctional. Why is it that these T cells are not able to, or why is it that um, um, organisms that don't have a thymus uh, cannot modulate their immune response? Is it possible that this particular mutation is affected in the T cells that we're talking about, in the T regulatory cells? Is this how can this particular mutation explain how T cells discharge their functions or not? Because the absence of this particular class of T cells actually causes an autoimmune disease, which is what is happening in scurfy mice and patients with IPEX. They wanted to check to see if this particular mutation is happening in the T cells, in the T helper cells. And when they did the analysis of the genome, they actually found that this same mutation exists in T helper cells that are dysfunctional, that cannot discharge their duty properly. Right? Right. So the T helper cell, and this is right. So the T helper cell recognizes a certain pathogen and sends a signal to the cytotoxic T lymphocytes and other T helper cells to sort of recruit them to the site of infection or whatever. The T regulatory cells, what they do is that they make sure that the immune response doesn't go out of hand. So if the immune response is not commensurate to the size of the infection, they will cause a suppression of the activity of the T helper cells and the cytotoxic T cells. They'll actually sort of put a brake on it. It's not necessary. Um, like a feedback, yes. There's some degree of feedback, I'm sure, because the communication happens through chemicals called interleukins and cytokines and other chemicals. So once the T helper cell recognizes a certain pathogen, it will release chemicals that other T helper cells and cytotoxic T lymphocytes can recognize, and they can flow in the direction of the infection. They know it's a chemotoxic, it's a chemotactic signal. Yeah. And so essentially, I guess that was the question. So would is it possible that in organisms, that in animals that cannot manage or do not have that capacity of regulating the immune response, is it possible that this particular mutation is preventing the T regulatory cells from discharging their function, and thereby allowing an autoimmune disease to happen, right? Gerard? Okay. I'll I'll go over it again in some parts. So that was one of the questions they asked, and so they found that yes, it is actually the same mutation that was observed in the scurfy mice and in the patients suffering from IPEX, that is occurring in the T regulatory cells, essentially. So in a sense, what that protein does, it's called a transcription factor, right? It's called a transcription factor. In other words, that particular gene in which the FOXP3 gene in which the mutation is happening is responsible for the development of that particular T cell into that particular T regulatory cell. That developmental program is not unfolding in that particular T cell because that particular mutation is present. So the T regulatory cell is not, in fact, being formed in the lineage that it should, right? And so this particular mutation in the FOXP3 gene is actually preventing that execution of the developmental program and preventing the T cell from becoming or maturing into a T regulatory cell. And therefore, if you have that mutation, it's the central the central tolerance system might still be working, but the peripheral tolerance is not going to happen because the immune cells, they just can't form the T regulatory cells. And that is enough for an autoimmune response to happen, or that is enough to allow an autoimmune response because there's nothing to prevent it. It's also not true that, you might say that it's not just the absence of this that's causing autoimmune, but also the presence of some other rogue cell, which do occur. So it's very likely that there is some rogue cell that's present and is not being stifled by the T regulatory cells. So why is this important, right? Why is it so important to know what is actually protecting our body from an autoimmune response? Why is it that why is this knowledge so fundamental, right? For a couple of reasons where you can find its application. One is that if you know this, maybe you can reconstitute the T regulatory cells in an individual who's suffering from an autoimmune disease and thereby mitigate the effect of that particular disease, right? So it has very potential therapeutic application. And this is not like Brave New World. This is actually this is something that can be done. You can take a population of T cells from the individual. You can influence the formation of a T regulatory cell by artificial stimuli, and then put those cells back into the individual, and therefore protect the individual from an autoimmune disease. Right? So that's one application that's straight up. The other problem is you can also prevent the immune system from rejecting a graft or a transplanted organism, it's transplanted organ, where if the T cells are recognizing the new organ as foreign and destroying it, then you can increase the number of T regulatory cells in the region of that organ and protect it from being destroyed. And you can cause a local increase in the concentration of those T regulatory cells by targeting them using these name tags to that particular organ. So you can increase, so that those are two applications straight off that we need such systems for. Right? And the third, the T cells can be made to target, or the T regulatory cells can be made to localize to the region of that organ using specific protein tags. How do you go in that direction? Right. So so the way the T cells, any T cell, can recognize a self, is through another receptor or another protein present on the cell surface of host cells. We call it the major histocompatibility complex. So it's called MHC. Right? So as soon as the cell sees that particular tag, it knows it's a self cell. Right? But obviously, assuming it's a normal cell, normal T cell. Right? But also, each organ or cells in each organ and each tissue have their own unique tags, so to speak. So by which certain chemicals in the body can localize to that particular. For instance, in breast cancer or in breast cells, there's something called the EGFR, or the epidermal growth factor receptor. So even though that hormone is being secreted, that growth factor, by the brain, it's only going to target the cells in the breast because only those cells have that particular receptor. And so each tissue has its own. And so this is one way of by which the body can send a signal from one part of the body to another, specifically like endocrine hormones, right? Endocrine hormones. That's one. So you can, in the same way, you can artificially create a group of T regulatory cells that can target a certain organ or stay in the vicinity of that organ, and ward off the immune response being launched by the other parts of the immune system, and protect the organ from being rejected, or at least rejected right away. You can, yeah.
Just curious, if they also run it in reverse, or if it even makes any sense, where it's possible to remove the regulatory T cells,
yes,
or where there is cancer, for example, and
I was coming to that. So that's exactly right. So that's actually the third really big therapeutic application, because what happens in cancers, when you have a tumor, they maintain a large number of T cells. They're infiltrated by a large number of T cells, obviously including T regulatory cells. And so if you can deplete that region of these T regulatory cells, you can cause the immune system to launch an attack, a much greater unmitigated attack on the tumor. So essentially, this is part of what now constitutes immunotherapy as well, where you're training the immune system to go hit cells that were originally your own cells, right? And so yes, it's still not known why, because what happens in many of these tumors or cancer cells is that these genes are mutated. So the proteins that are being synthesized from these genes are actually not self anymore, in the sense they don't belong to the normal body. They're actually rogue or not abnormal proteins. So it's still not known why the immune system doesn't recognize these mutant proteins as non-self and go attack them. But we do know that a lot of T regulatory cells exist in that particular region of the tumor, and I'm assuming that sort of mitigates the effect of the immune system on it. So if you can train the immune system to sort of treat that as foreign, you can kill it. There have been some major successes. Actually, immunotherapy is actually being used, I think, in prototypical fashion. Then one of the most beautiful things about it is that it is precision medicine. So the drug is targeting only the cancer cells, and it's unlike traditional chemo, which affects all fast-growing cells like hair or nails, and so it doesn't cause as many side effects. And so this is it's a really, if we can get it to work on a larger scale, uh, I think that'll be fantastic. I think the immune therapies that are now available are not exactly this. They come from a different angle, and so they also sort of, but it works. I mean, in principle and in preliminary studies, it seemed to work very nicely. Actually, even in therapy now, some of these are being allowed as therapies as well, very gently, very slowly. Because the problem is you don't even know, are you causing a local change? But in a system, at the system level, there's nothing like a local change. Every local perturbation has a very global perturbation. So we don't know what else is being affected. When you make that local change. So this was a paper where he showed that it's a mutation in the FOXP3 gene that alters or the control of the regulatory T cell development. And so it's preventing the T cells from maturing, or the regulatory T, or the immature T cells from maturing into a regulatory T cell, because the transcription factor is dysfunctional, and therein lies the base. So Sakaguchi was able to tie together the discoveries from Scurfy and IPEX and show how the class of T cells that he had identified were actually affected by the same gene. So he brought it all together. So that was really fundamental. And I think I have one last slide which you can just see and glance at and leave. But that was essentially the timeline. So it spans, I think, 50 to 60 years of work that has gone into identifying and finally coming to 2013 or so, where they identified that, they published that particular paper that you just saw. Went from 1959 to 2010. I just wanted to emphasize that again, that research is slow and good research is slower. But thank you all again for coming by. Very happy to. Thank you. Thank you.